This is the first time in our symposium that we are allowing grad students to present their research. And it is my pleasure to introduce our Iowa State University speaker, Chantal McCabe. She is originally from New Prague, Minnesota. She obtained her bachelor's degree in biology at the University of Minnesota Duluth. She came to Iowa State University to pursue the degree in plant breeding and genetics as a PhD student in 2011. Her project leverages traditional and cutting-edge approaches to attack the agronomic problem of brown stem rot in soybean. She is currently combining traditional genetic studies, plant pathogen interaction analysis, and genomic technologies such as RNA-seq and virus-induced gene silencing. Chantal is also a very active and enthusiastic student in our plant breeding program. Please, welcome Chantal McCabe.
Thank you, Betsa, and thank you for the organizing committee for inviting me to speak here today. Today I'm going to talk about a portion of my research titled Characterization of the Resistant and Susceptible Responses to P. gregata in Soybean. Soybeans are a really important crop. In 2014, in the U.S., there were 83.7 million acres of soybeans planted. The U.S. is the leading producer of soybeans, followed by Brazil, and soybean yield is continuing to increase every year. In 2014, the average yield was 47.8 bushels per acre, and the value of soybeans in just the U.S. was over $40 billion. So soybeans are really economically important, but we're still losing a lot of yield due to pests and pathogens. And just in 2014, the yield loss due to brown stem rot, the disease we're going to focus on today, was over 13 million bushels. And this number is probably a lot higher because we have difficulties phenotyping this disease. Brown stem rot is caused by the fungal pathogen Phialophora gregata, and there are two types of P. gregata, type A and type B. Both types cause internal stem browning. Just like the name brown stem rot implies, if you cut the stem open, the inside is brown, dead, and rotten. Type A also produces foliar symptoms, leaf necrosis and chlorosis, where type B has no foliar symptoms and the leaves look healthy. So with both type A and type B, you have a yield loss, and with both types, there are difficulties phenotyping. If you have type A in your field, you'll see leaf necrosis and chlorosis, but you might not associate it with brown stem rot unless you cut the stems open, because these foliar symptoms look identical to that of sudden death syndrome, another big soybean disease. If you have type B in your field, the leaves are going to look healthy. You won't see any symptoms on the plants unless you cut the stems open, but you will have a yield loss. So with brown stem rot, there's about a 40% yield loss. There are 3 resistant genes, RBS1, RBS2, and RBS3. And these are all dominant genes that have been mapped to chromosome 16. All of these genes provide partial resistance to brown stem rot. So even if you have a resistance gene, you're still going to have some symptoms. P. gregata is an imperfect fungus, meaning that it has a simple life cycle. There's no sexual stage that's ever been recorded, so there are no spores or no long-term survival mechanisms. So even if you have a resistance gene, you still could have some symptoms. It's an imperfect fungus, so there's no long-term survival mechanisms. So the best way to manage the disease is to use crop rotation and tilling, practices that get the residue out of the field, and then also to use a cultivar with a resistance gene. So my work has focused on brown stem rot resistance in grad school. I've been using multiple different approaches, classical genetic studies to look for new sources of resistance, microscope studies to look at the plant pathogen interaction, and then also genomic technologies like RNA-seq to look at differential expression of the genes. And today we're going to focus on that last portion, the RNA-seq work. So there are 3 resistant genes, but we don't know a lot about how they work or the defense networks regulating the response. So we want to ask questions about expression. Are the resistant and the susceptible genotypes turning on different genes? If they're turning on the same genes, are they turning them on at different levels? And what are the differences in the leaves, the stems, and the roots? So in this experiment, I had 2 genotypes, a resistant line and a susceptible line. The resistant line was PI 437970, which has the RBS3 gene. and the susceptible line was Corsoy 79. And half the plants in each genotype were inoculated with the pathogen, P. gregata, type A of the fungus, so stem and leaf symptoms, and half the plants were inoculated with water agar as a mock-inoculated control. And 1 week after infection, I collected leaf, stem, and root tissue from the plants. This is important because we normally don't even phenotype for this disease until 5 weeks after infection. The plants are already 7 weeks old. So if we could identify signatures of defense response sooner, we can really advance the screening process and advance brown stem rot breeding. So to do an RNA-seq experiment, we first need the RNA. This RNA gets extracted, we sequence it and map it to the soybean genome. And we use the Williams 82 reference genome for soybean. In this step, we have a lot of quality control that happens and data cleanup. We're then able to analyze the expression with different statistical software packages. And when we analyze the expression, we look for differentially expressed genes. So in this picture, the brown dashes are reads from the susceptible line, the green are reads from the resistant line, and they've been aligned to the Williams 82 genome in blue. So gene number 1 is expressed in the resistant and the susceptible, so it's not differentially expressed and not something we're going to pay attention to in the analysis. Gene 2 is expressed only in the susceptible, gene 3 is expressed only in the resistant, and gene 4 is expressed in both genotypes, but it's expressed more in the resistant. So genes 2, 3, and 4 are differentially expressed, and they're the signatures of expression that we're looking for when we're analyzing RNA-seq data. So for every differentially expressed gene, we can generate an interaction plot. And these interaction plots help us check the quality of the data and visualize the data. So in all of these plots, treatment is on the x-axis, mock-infected and infected. The y-axis is the expression data. On every graph, there are 2 lines, a blue line and a red line. Blue is the susceptible and red is the resistant. So in the first picture, it's showing a treatment effect, differences between treatments regardless of the genotype. So the 2 lines are on top of each other, so the treatment is affecting both of the genotypes. In the second picture, it's a genotype effect. So differences, this gene is being differentially expressed between genotypes regardless of the treatment. So the blue line, that susceptible line, is always higher no matter which treatment it's under. And the third picture is a genotype by treatment interaction effect. So the genotypes respond differently depending on which treatment they're under. Another way to visualize this RNA-seq data is with a program called MapMan. So every one of those colored boxes is a gene being expressed. And this first picture is what happens when the resistant plant is infected. And there are a lot of genes being expressed. And a lot of these genes are related to defense response as well. MapMan is grouping these genes into different functional groups. So there are genes related to hormones, related to signaling, related to abiotic stress. A lot of genes are being expressed. Compare that to the second picture when the susceptible plant's infected, and there's a lot less going on. The resistant plant is expressing more genes than the susceptible, which answers that first biological question that we had. The resistant and the susceptible plants do different things when they've been infected. And to answer our other questions, if they're expressing the same gene, are they expressing them at different levels, we use heat maps. So this heat map was made with almost 7,000 differentially expressed genes due to genotype, differences in the resistant and the susceptible. And this heat map is based on a z-score, which is individual gene expression of the sample compared to the mean gene expression of all those samples. So if an individual gene is expressed in white on this heat map, it doesn't deviate from the mean expression of the samples. If it's shown in yellow, it has a mean higher than the average of the samples. Brighter yellow is an even bigger deviation. If the individual gene on the heat map is blue, the average is less than the average of expression of all samples. Darker blue is an even bigger deviation. The first thing to notice on this heat map is that all of the genes cluster by tissue. So the stems and the roots are closer related to each other than the leaves are. And within each of these tissue clusters, we're also clustering by genotype. So the susceptibles are more similar to each other than the resistants are. And it's really clear in the upper corner. The susceptible, infected, and susceptible mocks appear yellow, in the leaves when the resistant infected and the resistant mocks appear purple. So regardless of the infection, the resistant and the susceptibles are responding differently. The other axis on this heat map is clustering by expression. So we're clustering all of these genes by similar expression patterns, and we ended up with 8 different expression clusters. So one of those clusters is this blue cluster, and we can see that they're all similar expression. So the leaves are all expressed in one direction, they're yellow, and the stems and the roots are expressed in the other direction, they're blue. So to figure out what these clusters do, we look at their gene ontology, or the GO terms. And gene ontology lets us put all those genes into different functional groups. And every one of these clusters correlates to a distinct biological function. So the first cluster has genes related to DNA replication, cell proliferation, and gene silencing. The strongest genotype effect that we see is happening in the leaves. The upper corner, where the resistant infected and mock infecteds are one color, and the susceptible infecteds and mock infecteds are the opposite color. So regardless of infection, the genotypes are doing different things. They're expressing different genes. The biggest cluster that we see is related to wounding and defense response. And every resistant plant in this category is expressed differently. The resistant leaves are expressed stronger than the susceptible leaves. The resistant stems and roots are expressed in blue, where the susceptibles are expressed in yellow. So genes relating to wounding and defense response are either expressed more strongly than the susceptibles or in the opposite direction as the susceptible plants are. So this tells us that even not worrying about treatment, the resistant and the susceptible plants differ in how they respond to infection, differ in their innate defense responses. These genotypes are predisposed to just do different things. The next heat map is made with almost 4,000 genes differentially expressed between treatments, differences in the infected and the mock-infected plants. And again, this heat map clusters everything first by tissue. So the stems and the roots are again more closely related to each other than the leaves are. But now within each tissue, things are clustering by treatment. So now the resistant mock leaves look more similar to the susceptibles than they do to the resistant infected leaves. That one band of resistant infected leaves really stands out. Again, the heat map was clustered by expression, so genes with similar expression patterns are grouped together. We have 7 different expression clusters. We can look at what these genes do by looking at the gene ontology again. And the largest category is DNA replication, cell proliferation, gene silencing, and the regulation of the hypersensitive response. So the genotype effect that we see, or the treatment effect that we see, is happening in the leaves. The resistant infected leaves are blue, when the rest of the susceptible treatments are in yellow. So when they've been infected, the resistant plant is responding differently. The brightest colors that we see on this heat map are both seen in that regulation of the hypersensitive response, where the resistant plant changes direction of expression, and also in photosynthesis, where we have a brighter yellow, so the resistant plant is expressing them to a greater degree. So this heat map is showing that even the same gene can either express things to a greater degree due to treatments or in the opposite direction. We also see hormones being differentially expressed. And a lot of these hormones are defense-specific, like jasmonic acid, ethylene, and salicylic acid. And then we were also interested in looking at what was happening in that RBS3 region. RBS3 is one of the resistance genes for BSR, and it has been mapped to chromosome 16. So in that RBS3 region, there are about 40 genes, and about 20 of them are differentially expressed in my data set. Everything on this heat map that's in blue is expressed more in the resistant. If it's yellow, it's expressed more in the susceptible. So what stands out is that dark blue column in the roots. And we didn't see a very big treatment effect in the roots, but there is a big genotype effect in the roots, especially in that RBS3 region. And this dark blue area in the roots, they correspond to RLPs, or receptor-like proteins. And this is a special class of disease resistance genes that has been known to provide resistance to fungus in both apples and tomatoes. And this big differential expression is happening just 1 week after infection. We can't even see a phenotype at this point. So what we wanted to do was look for SNPs. SNPs would allow us to develop molecular markers to help screen for this disease earlier. And in just that RBS3 region, we were able to find 471 novel SNPs. So with this computer program, the top panel is the resistant plant, and the bottom is the susceptible plants. And there's a polymorphism located right here, where there's a T present in the susceptibles instead of the A. So these 471 novel SNPs can be used for developing molecular markers and also for fine mapping this RBS3 region. So in my work, we've seen that there's a large effect on both genotypes and treatments 1 week after infection, which is a lot sooner than we can identify by phenotyping. We know that the resistant and the susceptible lines respond differently to infection. In general, the resistant plants are expressing more genes than the susceptibles. And we can cluster all these genes by expression pattern, and all of these clusters correlate to biological functions. And we were able to identify 471 novel SNPs just in that RBS3 region, which we can use for fine mapping and for developing molecular markers. And we're currently using virus-induced gene silencing to characterize those candidate RLP genes in that RBS3 region. So I'd like to thank you all for listening today, and thank you to my co-major professors, Dr. Graham and Dr. Cianzio, my committee members, Leonor Leandro, Dan Nordman, and Danny Singh, and all of the help and support I've gotten from the Graham Lab, Cianzio Lab, O'Rourke Lab, and the Whittam Labs. And also thank you to my funding, the Agronomy Department at Iowa State, USDA, NCSRP, and the Iowa Soybean Association. Thank you.
All right, we do have time for a couple questions. So again, reminder, please use the microphone. Thank you.
Thank you for a really excellent presentation. How often do you find brown stem rot and sudden death syndrome co-infecting? And also, is there any cross-resistance between the two, the resistance genes in the two diseases?
I'm not sure how often they're found in the same field, but I know it does happen. And as far as cross-resistance, I'm not sure.
Okay, fair enough.
Chantal, great presentation. I was wondering that you have the leaf and stem and root, three tissues. In the literature, this type of disease, when it happens, is there any tissue that you do not expect any difference between those resistant and susceptible? How that can be factored into your analysis? It's a generic question.
So we're hoping to find that answer out. In nature, the pathogen enters the plant through the roots, but in the lab, I infect in the stems. So we harvested the three tissues and are analyzing them just to see what the differences are, to see if there's communication happening between what's below ground and above ground, and how all of those tissues interact. But there hasn't been a lot of work done on this previously.
This is not a question for you, Chantal. This is just a comment for Paul. What you asked Paul was not really part of Chantal's dissertation, but we will have information on the interaction between brown stem rot and sudden death syndrome, because we have developed populations and recombinant inbred lines that have the two sources of resistance. So shortly, in another research, we will have some information about the interaction.
All right, well, let's thank Chantal one more time, and we'll